topic immunological techniques. In that the technique we are going to see is ELISA, right? So you should know what is mean by immunological techniques, right? It include both experimental methods to study the immune system and methods to generate or use immunological reagents as experimental tools. The most common immunological methods related to the production and use of antibodies to detect specific proteins in biological samples. Here you should know what is this proteins in biological sample. It means that is the antigen. What are the specific antigens present in the biological samples that will be detected by these immunological techniques, right? Then we should know what are the recent immunological methods. In earlier days, common agglutination, heme agglutination, immunodiffusion methods and immunoelectrophoresis, all that did for the detection of antigen present in the biological samples. In the medical field, nowadays the recent technologies were applied to detect the antigen, that are ELISA, RIA, immunofluorescent immunoassays, hemoluminescence immunoassay. These are the recent technologies adopted in the field of medicine and to detect the specific pathogen or antigen or in the form of proteins present in the biological samples. Okay. Then you should know what is mean by ELISA. ELISA, it is the enzyme linked immunosorbent assay. ELISA means the expansion for ELISA is enzyme linked immunosorbent assay. So it is a plate based technique used to detect and quantify peptides, antibodies, proteins and hormones. So this ELISA is the basic assay technique known as enzyme linked immunosorbent assay, also referred to as EIA, right? Enzyme immunoassay that carried out to detect and measure antibodies, hormones, peptides and proteins in the blood. Okay, in the blood, that is what are the antibodies present in the blood, hormones, the different types of hormones, peptides and proteins. Okay, then the antibodies are blood proteins produced in response to a specific antigen. It helps to examine the presence of antibodies in the body in case of certain infectious diseases. Okay, what are the antibodies? Okay, so that will be help to examine the certain infectious disease, right? So these are the basic, this slide is going to tell about basic steps of ELISA, right? So I said already, ELISA, it means enzyme linked immunosorbent assay, right? So first step is antigen of interest is absorbed on the plastic surface. So that is known as sorbent. Okay, the first step is the diagram is showing that in that arrow mark, first step in the bottom. Okay, so the antigen of interest is absorbed on the plastic surface, that is called sorbent. Then the second step is the antigen is recognized by specific antibody, that means immuno, right? So that is the expansion they are giving, right? So the second step is antigen is recognized by specific antibody, that is called immuno. Then this antibody is recognized by second antibody, so that is also known as immuno, which has enzyme attached. So that's why it is called as enzyme linked, right? So this antibody, the secondary antibody which is connected with the enzyme, so that is enzyme linked. Then finally fourth step is the substrate react with the enzyme to produce a product, usually colored form. Okay, so the the fourth step is substrate is added. This substrate reacts with the enzyme to produce the products, usually in the form of colored. So the colored product is measured as a assay, right? So in the form of the quantifying the antigen present in the biological samples. Okay, so the colored product is equal to measure of antigen present in the biological sample. So these are the steps used in this ELISA, right? And then these are types of ELISA. In the ELISA, there are different types of ELISA. So one is direct ELISA, the first one, and then second one is indirect ELISA, and third one is sandwich ELISA. Fourth one is competitive ELISA. Okay, so that is the first directly only one antibody, the direct ELISA, and then antibody with the substrate. Okay, so that is the indirect. Then sandwich. So each one will detailedly seen in the next slide. Okay. So first one is we will see about direct ELISA. Here the solid state, the first we are adding is antigen. The antigen is coated onto well by passive adsorption, right? The second step is antibody conjugated with enzyme is added and incubated with antigen and incubation, right? So this is the adding antibody. Second step is adding antibody and then adding enzymes, then there will be the incubation, right? Then third step is adding substrate, or and the substrate called as here chromophore. Here the substrate name is chromophore is added. Then there will be the color development. So this is the direct method of ELISA, right? Then we'll see about indirect ELISA. Okay, so in the indirect ELISA, first step again, the antigen is coated onto wells by passive adsorption and then incubation. Then second step is there will be the adding of primary antibody. Primary antibody is added and incubated, right? Then third step is anti-species antibody. Okay, anti-specific antibody conjugated with enzyme added and incubated. Okay, so fourth step is substrate is added. Then there will be the color development. So this is the indirect ELISA, right? So this is third one, that is sandwich ELISA. Here we are adding first capture antibodies. There is the difference between these three two, that is direct, indirect and sandwich. In this sandwich, first we are adding capture antibody. So that is the difference. Then we are adding the antigen, right? Then detecting antibody, then secondary antibody we are adding, then enzyme, finally we are adding the substrate, then there will be the color development. Okay, so that is the sandwich ELISA, right? Then fourth one is competitive ELISA here. Okay, so what will happen in the competitive ELISA? First step is unlabeled antibody is incubated in the presence of its antigen, that is the sample. Okay, so first we are adding unlabeled antibody is incubated in the presence of its antigen, that is in the form of sample. Then these bounded antibodies or antigens complexes are then added to an antigen coated well, right? Then the plate is washed to unbounded antibodies. There will be the in each and every step in the all three ELISA methods, each and every step there will be the washing step. Okay, so that will do the unbounded antigen antibodies will be taken out. Okay, so for that there will be the washing step in each step. Okay, the plate is washed, so unbound antibody is removed. Okay, so that is more antigen in the sample, the more antigen antibody complexes are formed, and so there are less unbound antibodies available to binding of antigen. So hence that will be the competition between the antigen antibody binding. So there will be the competition between binding between the antigen antibody. So that is the competition. So that is why called as competitive ELISA here. Then the secondary antibodies specific to the primary antibodies, there will be the addition of secondary antibody to the specific antibody and is added. This secondary antibody is coupled to the enzyme. In the fourth step we are seeing, that is in the third we are adding the we are adding the antigen, okay, then we are doing the washing step. Also the antigen antibody bounding will be happen. In this washing step, unbound antigen antibodies are removed, okay. Then the fourth step is the secondary antibody added specific to the primary antibody, right. The secondary antibody is coupled to the enzyme, right. Then fifth step is a substrate is added. Here the substrate, substrate it means chromophore, okay. So a substrate is added and remaining enzymes elucidate that is in the chromophore, right. Then the reaction is happened, right. The reaction is happened, the color developed. In between there will be the reaction stopped to prevent eventual saturation of signal, that is there will be the signaling, so that time the reaction will be stopped, okay. Then the colored, the intensity of color will be measured and antigen antibody quantification will be estimated. So okay, so that is the competitive ELISA, right. Then so we have seen what is ELISA, okay, what are the ELISA methods we are adapting in older days and then the recent days, okay. Then individually we have seen what is direct ELISA, what is indirect ELISA, what is sandwich ELISA, and what is competitive ELISA, okay. So we have the each and every steps involved in these ELISA types. Here we have to know the what are the advantages of these ELISA types, and then what will be the drawbacks and disadvantages of these types of ELISA, okay. First we will see the advantages, okay. So in that the direct ELISA, right, so that will be rabbit, okay. The direct ELISA, rabbit, it means speed, and then the secondary antibody cross reactivity eliminated, okay. Secondary antibody cross reactivity eliminated. Then in meanwhile we will see the disadvantages also, but the drawback is only low sensitivity. Here the low sensitivity, and then specific antibody for such ELISA time consuming, and then it is the time consuming process, and then expensive, more expensive also, so that is the drawback. And then we will see the indirect ELISA, okay. So in the indirect ELISA, the advantages is high sensitivity, cost saving, and flexible, and can use many primary antibodies, okay. So that is high sensitivity, cost saving, flexible, more flexible, and can use many primary antibodies, okay. Then the risk is the risk of cross reactivity between secondary antibodies. We may not correctly estimate it because of this risk of cross reactivity between secondary antibody, okay. Then sandwich ELISA, right. So here the application is minimal sample purification needed. Here the purification can be minimized, and then high sensitivity and specificity. Here also high sensitivity and specific, very specific to the antigen antibody. Then the drawback is must use matched pair primary and secondary antibodies, okay. The pair should be clearly noted, that is between the primary and secondary antibodies, and also it is the time consuming process and expensive also. Then we will see the advantages of competitive ELISA, that is minimal sample purification needed. Here also we can the purification step can be minimized, okay. Minimal sample purification needed and used to measure large range of antigen to a sample, right. And then used for a small antigen. Even we have isolated a small amount of antigen that can be measured with this competitive ELISA. And then low variability, okay. The variability can be very, very less, right, than in the readings, okay. So that's why they are saying. Then the disadvantages is low specificity, so carried by you to use a dilute sample. Okay, the sample can be, even if a small amount of sample that can be diluted and then we use, so that lead to a low specificity of the detection. So that is called as this drawbacks. Okay, so these are the advantages and disadvantages of these types of antibodies. So you should understand what is the types of ELISA and then the advantages and disadvantages. Based on that, you can use this technology in your further studies. Okay. And then you should know the general applications. Okay, in the ELISA, it is the advanced technique. ELISA is the advanced technique. So it is mostly used in medical field. Okay, so for that, what are the applications applied in this field? One is serum antibody concentration can be detected, right? Serum antibody concentration can be detected. Then detecting potential food allergens. So that will be one form of pathogens. Okay. So for detecting potential food allergens like milk, peanuts, walnuts, almonds and eggs. Then disease outbreaking and tracking the spread of diseases. Okay, we can detect this technology using where will be the outbreak is increased, right? And then we can track the range from where here to here, where the diseases, what are the diseases are increasing, like that. Okay, HIV, bird flu, common cold, cholera, STD. Then detection of antigens, then detection of antibodies in blood samples. So these are the applications of ELISA, right? Okay, girls. Thank you.